Okay, welcome everyone to the Longevity Acceleration podcast. We're super excited. For today's episode, we're speaking with Jean Hebert. He's a professor of genetics and neuroscience at Albert Einstein College of Medicine in New York, and also the founder of BE Therapeutics, a really awesome startup in the replacement strategy for aging. Welcome to the show, Jean.
Yeah, welcome.
Thanks for having me. Always a pleasure.
So, John, I think one of the things we wanted to start off by asking all our guests is basically getting a sense of what their ultimate goal is. Because, as you know, the show is called Longevity Acceleration Podcast, but the word longevity means many different things to different people. So, we just wanted to get a sense of what's your goal? What's your ultimate goal?
What it comes down to is, I enjoy life. And even when I'm not enjoying life, I prefer it over the alternative. And so, that's ultimately my goal is to stay alive.
I agree. That's a noble goal. And I think most people in our community at Longevity Biotech Fellowship also share that same goal, that same mission. The reason why we started this podcast series was because we had so many people who share the same ultimate goal. And, our organization, Longevity Biotech Fellowship, the LBF, tries to help people find ways to have a significant impact on achieving this goal through their careers or their work or whatever. But it's never exactly clear what are the main ways for us to actually to allow us to continue living, to extend lifespan, ideally indefinitely. So with LBF, we've tried to create this roadmap, this narrative of the different ways that technologies could be developed to allow people to live indefinitely, to solve aging completely, essentially. Obviously, replacement is one of the major themes, one of three major pathways strategies that we've identified. John is obviously one of the foremost experts on the field. You wrote a book, Replacing Aging, which was really formative for me in my thinking about the problem too. But in terms of the strategy of replacing aging, maybe you can walk us down a little bit through memory lane, because I remember the last time Mark and I talked to you about the roadmap and replacement, your background is a professor of genetics. One of the things you mentioned was when you were first early in the field, you were looking at different ways to try and attack the aging problem, and you actually changed your mind. So maybe you could tell us a little bit about when that was and how you were thinking about aging back then, like your mental model, and what made you change.
I said this before, but even when I was a little kid and I found out we grow old, fall apart, and die, I was not happy. And after getting over my depression for a couple of years in grade school, I started reading science, like how could we fix ourselves? In high school, I guess, I was excited to know that other people were interested in understanding aging. Back then, someone had identified a protein that accumulates damage with age in circulating blood. And it was like, oh, that's really exciting. Like maybe that's part of it. Maybe we should target that. And, I kept reading and learning. A book that had a big impact on me was Caleb Finch's book. I forget what it's called, something longevity and senescence, longevity and the genome or something. And in there, it's like this pretty thick book, and it covers fairly comprehensively what was known at the time, which, was probably 2, 3 decades ago already, what was known about the aging process. And already in there, it became clear to me that, this is like a hugely difficult problem because of the damage that occurs at the molecular level, mostly outside our cells. So early biochemists were studying abundant proteins like collagen, which makes up the bulk of our bodies, other big proteins that, don't turn over much, if at all, during our lifetimes, and described these complicated forms of damage. And a lot of them gave up on possibility of beating aging because of that. Because that damage was very complex, very stochastic, non-enzymatic. So it's not a biological process. It's really a physical one of degradation over time, just like everything else in the universe. And so trying to solve it with some approaches, it just won't work. But for a time, I thought genetics could still do it because it's the code of life. And I thought, maybe we can encode better things, more robust life in a way. And so I went into genetics, but still reading at the time and realized that it was going to be difficult and that maybe a better way is replacement. At first, I was thinking replacement of cells might be good enough. For a long time, I was thinking about how you could replace cells, but always knowing in the back of my mind that there's still a problem with the extracellular environment of those cells that's getting old. And so I found my way to the inevitable conclusion that if we're going to do this in any reasonable amount of time, we needed to replace whole tissues. And what's the most important tissue, I think, is the brain, and within the brain, the neocortex. So I started focusing on that and how we could replace tissue in the neocortex, possibly still thinking about how we could reverse some of the damage to buy time, but eventually gave up on that because that seemed way more challenging than actually just growing new tissue. But there were also two turning points for me to come to that conclusion that it would be easier. One is the realization of just how our neocortex, our brains, encode information, which is super plastic. So information, memories, our self-identity can move from one part of the neocortex to another over time. And we've even observed this in the clinic in humans of advanced age. And so it's really remarkable because of different lesions that they have, for example, slow growing gliomas. So that really meant that, okay, so maybe if things can move without a loss of continuity of self, uh, then we could maybe remove old tissue and replace it with new tissue. And the second thing that was pivotal in realizing that this was the best approach for the brain as well was that if you put in young brain precursors, they seem to not care that they're in the adult environment, and they'll project long distances to normal targets and integrate with existing circuits. And this is a super robust finding in the field. Dozens of labs have reproduced it. So those two things together, like the plastic nature of the brain and how it encodes information and the ability to add new tissue, really was to me like the moment where, okay, we can beat aging and we can do it in a reasonable amount of time if we mobilize. And so since then, I've been really excited by that. And to some extent, also a bit frustrated at how slow it's been to mobilize and get that job done.
Yeah, that actually leads into a question I was going to ask. I went through a similar series of thinkings about my approach that evolved over time, but there's certain dominant narratives out there, right? Like they tend to come and go. Like there was telomeres, there was senescent cells, there's, I think Aubrey's damage repair narrative has been probably the most well-known one. I'm kind of curious about the timeline. When exactly was it that you realized that this sort of uh, body and gradual brain replacement was the most likely to succeed in your view, and why? I guess you wrote your book. What year was that released? But also, what year did you find out about this and it? And also, just like, why has nobody heard about this? Because it seems like even when we talk to people within the field, most of them still don't know about it.
Yeah, I mean, to some extent, it's intuitive. The notion of replacement is intuitive, and it's been around for a long time. I remember early 2000s going to a conference. It was a sense research conference at the time. And someone just came to me afterwards and said, "Why don't we just replace the whole body?" This was not a scientist, but it's just very intuitive that this is an approach that will work. And I'm like, I don't know why we're not doing that, because there is the problem of the severed spinal cord, but there are currently ways of dealing with that. So yeah, why aren't we doing it? Yeah, I started being focus more on the brain. Even at that time, I started studying brain development for the purpose of recapitulating development of the brain in the adult in the mid 90s. I was already committed to the brain and didn't want to focus on, or didn't have the bandwidth to focus on anything else at the time. Yeah, I mean, I guess it was sort of progressive to some extent when I got to that moment where replacement was going to allow us to beat aging. I mean, I guess different moments, like the ones about brain tissue replacement, how we could do it without loss of function or continuity of self. That was probably 2015, 2016. If you read my book now, I feel a little embarrassed by it because my thinking has progressed quite a bit for all the three major parts of the book, like why non-replacement approaches are even more challenging and require technological breakthroughs that we can't imagine yet, and why the progress made in replacement has to some extent shown proof of concept with a lot more organ replacements going on and other technologies. And then for the brain, that's where I was more open to potential approaches other than outright progressive tissue replacement. And, since then, that has become much clearer to me that that is the most achievable way of reversing brain aging.
Awesome. Yeah. So I think we're going to dig into the details of brain replacement. But first, before we do that, I'd like to get your thoughts on maybe some of the other strategies, because you mentioned other strategies that you have discarded or at least decided not to focus on, not necessarily written off entirely, but decided that they're not your focus area. I had a similar realization when I was doing my PhD. I was like manually pipetting cells and spent 2 years showing that my predecessor's work was not reproducible. And I was like, oh my God, if this is the way we do research, it's just not solving aging in my lifetime is just not plausible. And so my reaction was, how can we make research go faster? Um, and so that's the type of work that I ended up shifting towards, first in technology development, and now working with Nathan to try to grow the field, both in terms of the number of people working on it and the amount of capital deployed to it. So that's the direction I went. But you mentioned the complexity of aging. Maybe just say a few more words on that, because you do describe it in your book, Replacing Aging, but yeah, you just said that you would maybe rephrase it, or you've even are more, that you have an even deeper appreciation for the complexity of aging than you did then. So maybe say a little bit about that, because I don't think people necessarily appreciate how difficult this problem is.
So I did, mention the fact that a lot of the damage that accumulates over time accumulates to these long-lived proteins and is non-enzymatic. So it's not a biological process. It's a physical process. Elastin, collagen are canonical examples, but by no means the only ones. And the types of damage that have been described for these that accumulate over time are not recognized. So for collagen, for example, when it accumulates damage, it can no longer be turned over by collagenase. And yeah, so that damage just keeps accumulating over time.
Like it's not like
Yeah,
us.
yeah. There's actually a fairly long list that's been described by these biochemists that looked early on at proteins like elastin and collagen. Um, and I guess what's also important is that now that we know like the whole genome sequence, we know what the genes are in the genome. We know there's no secret hiding genes out there that encode machinery that can actually do anything about this damage. There isn't any. So
Yeah, that to me is like the hidden assumption underlining people that are trying to do drug development, because the pharmaceutical approach is to just modulate existing machinery. But if there is no machinery to do the job, then there's nothing to modulate. Would you agree with
and
that?
oh, absolutely, absolutely. And it also is relevant for partial reprogramming, right? That's just changing gene expression to a more immature cell state. But still, there's no genes there that can deal with the damage that has accumulated to that point, or that can slow it down or recognize it or anything. And
Yeah,
so that's why
yeah.
that approach will fail as well. In and of itself is not going to do the trick.
Yeah, I think that's the key. And I think everybody thinks that it's going to be a combination, right? So the reprogramming by itself doesn't solve all the problems.
I don't even know that it solves any problem because, there's a fairly
Ooh,
solid
hot take.
There's
Well, it solves one problem. Where should I put all my money if I'm like a eccentric Russian billionaire?
Yeah, no, that's true. But yeah, I mean, I say that again based on a pretty solid literature of heterochronic transplants, or taking cells from young tissue and putting them in old tissue and show that they become like old cells in their behavior and their metabolism and what they can do. And the opposite is true. And maybe the opposite is also telling, that you can take old cells and put them in a young tissue and they behave like young cells. So really, this focus of the longevity field almost exclusively on intracellular processes is going to have limited benefits altogether. And that includes almost everything, or maybe everything, except replacement therapy.
Yeah. Well, I guess there's still one caveat here, which is just like, if we're playing devil's advocate, it's still this idea of, okay, maybe we don't need to solve aging completely, right? Like one of the elegant ways that replacement solves the problem is basically saying, yeah, we're going to solve the problem kind of all at once, and we don't need to know everything about aging. But I guess Aubrey's sort of introduction of the concept of longevity escape velocity, and it'll be combinations of things, and we have all these different categories, and then we just chip away at a bunch of them and put them all together, and then maybe that's a solution, and maybe you just buy enough time to get to the next bridge. Curious what you think about that, actually.
Yeah, it's just there's no evidence that any approach we've taken actually slows aging. Even in mice, there's very little evidence that anything works, much less in humans, where it may be more challenging as well. But the approaches that are described in addressing these different things, although to Aubrey's credit, he includes damage to ECM proteins, which are at least on the radar there. For many others, it's not, including the more popular hallmarks of aging. It's all intracellular. Other than that one thing of tackling the extracellular matrix as part of Aubrey's categories, the rest are intracellular. And again, I think that will have limited impact on age. And the approaches taken to target the ECM are maybe one tiny piece of it, which on its own is not going to have much of an impact. So unlikely. No one's been able to do that yet. But if they could, then it still wouldn't have, I don't think, much of an impact. And then doing it for all the forms of damage, without killing cells and tissue, if you do it chemically, for example, or if you do it enzymatically, again, that could have terrible side effects. It's very difficult for an enzyme to know, the difference between one good covalent bond and a bad covalent bond, for example, that looks similar. So likely to be bad side effects there. And now multiply that by 100,000 different targets that you need, and delivering these things to get there, again, without disrupting normal processes. It just all starts to seem like ridiculously difficult. And I won't say impossible, but that's what I'm thinking. It's just so much easier to replace tissue. And so all these approaches, some of them do show health benefits. And so we're talking about more compressed morbidity. So if you can solve all the health problems, great. Everybody is going to get into their 90s. But there's still that precipice there where things have broken down so much that you'll get a hemorrhage any day, or your days will be numbered. Um, so, I mean, but those health benefits are definitely worth pursuing. I think it's really important. I just don't like the confusion that those are slowing down aging, because there's really no evidence that they are, and there's no reason to believe that they are. They're just making us healthier as we age.
Healthy aging is kind of a oxymoron term that I think neither Nathan nor I are a big fan of, but that's sort of what a lot of the work is trying to do, is try to there's a sort of a potential lifespan that the human genome has, and a lot of people fail to meet it for various reasons. And you can focus on trying to help put more people reach that potential. But that's a very different, the strategies that you would employ, the research that you would do if your goal is to actually change the maximum, I think are very different. And so that's, I think, why it's important to highlight that. And like you say, I think the majority of the resources are going towards things that wouldn't affect the maximum or wouldn't plausibly affect the maximum. Although, again, yeah, we can challenge you a little bit more on the bioengineering side. I think one of the reasons for, well, so there's, for the replacement strategy, the proof of concept per se is that obviously new humans are born young, right? So it's obviously possible to generate young tissue by just making a new human. And so why not just replace the whole body, grow a replacement body? And then you've also been the person who's like made the strong case that this is all plausible for the brain as well, which is harder, but at least plausible. When it comes to the bioengineering approach, people like to say it doesn't break any law of physics. As long as you have free energy coming in, you can maintain order. And there's a lot of variety within nature already in terms of longevity. Like there are organisms that live longer than we do. And there's a lot of variety within like sort of closely related species. And by identifying so all of the mechanisms by which nature has produced variance in longevity, perhaps we could modulate those pathways. I think that's one of the arguments in bioengineering. I guess, yeah, so that's, and you looked at that as a genetics thing, but I guess the delivery problem is probably maybe, is your biggest concern the ability to actually make those modifications, or to even know what modifications to make, I guess.
Um, Certainly, you can make modifications like that ex vivo to cells. You can heavily modify genomes, and we've seen that with pigs having 60 genetic modifications, but that's with the cells before making the pigs
Yeah,
out.
germline modification. Yeah.
So it's doable. Doing it in vivo gets, yeah, it's the delivery of like CRISPR machinery to the cells you want. Again, I think it's very difficult. Like, we're already one of the longest lived species. We're not the longest. There are some longer lived species. I think what would be interesting, I don't know if anybody's actually doing this, but would be to look at their long lived proteins and how they differ from ours, and try to make those modifications and see if that has an impact. Because if it is indeed the extracellular long-lived proteins that are really killing us over time, which the evidence points to, then maybe targeting more of those would make more sense. But then how do you do it in vivo? And
Mm.
the only way I see right now that you could do that is by replacing tissue, right? Because you can modify the cells beforehand, make the new cells, new tissue, uh, and introduce those. But if you're doing that, then you've already solved the problem, I think. So
I think, yeah, analysis of the differences in the way long-lived proteins accumulate damage over time in different species. Sounds like a really good impetus grant proposal for any of our listeners out there, maybe. But yeah, like you say, you still run into the delivery problem. And I ask this of a lot of people because, like, for instance, even if you are a strong proponent of reprogramming, how are you going to deliver those mechanisms, right? Because it has to be cell by cell. I think we know this, that you want to be able to check the cell's state and then deliver the precise dosage of reprogramming factors for that particular cell type and that particular cell's state, which requires high payload and full body gene delivery. And that doesn't seem to be, I mean, for all the people that are working on reprogramming that I've asked, they don't have a solution to that problem. So it's kind of interesting to be developing a therapy that can only work in germline modification, I guess. So that I think highlights the importance of the gene delivery problem. And I guess if there was a major breakthrough in the gene delivery technology and gene delivery technology, would that affect your thinking? How much would that affect your thinking on this?
By major breakthrough, you mean being able to deliver exactly what you said, fairly large payloads
Large payloads, full body.
with specificity to cell
Yeah.
type.
Yeah.
Yeah, yeah. I think there, you know, I think you still have to think about, fixing the damage that's around cells. So that might mean in that payload, you've developed new enzymes that can now recognize this damage that don't currently exist. But at least, you've gotten over one big hurdle towards that. Yeah.
Okay. All right, so maybe we should move to your area of expertise and dig deeper on replacement and ask you some hard questions. So I've been talking for a little while. Nathan, what's your devil's advocate position on replacement? Let's see what we can push John on here.
push some holes. Yeah, well, maybe we should just start off with the basics for people who aren't familiar, so we're all on the same page. We've scoped this out a little bit on our website for anybody who's interested, longbiofellowship.org/roadmap. Looking at replacement, there's some materials there. But so Jean, just to agree with us, I'm guessing you like the whole strategy for, let's say, hardcore replacement versus like intermediate replacement or other types of replacement. For us, when we talk about replacement at LBF, we're talking about basically whole body replacement with some sort of cloning with the neocortex knockout combined with progressive brain replacement. So
like producing a clone that's non-sentient and using that as effectively a donor tissue, like it's a new body for you that was never sentient.
Yes, yeah. So that would solve aging in the body, and the brain is what you're working on, John. Yeah, so that's kind of like the strategy. So I guess I don't know if we want to speak too much about the body replacement, but we can come back to that later, which is really interesting topic. But before, I guess, yeah, I'm curious. Why did you choose brain first instead of the body? Because they seem like, probably both necessary at some point. Maybe, maybe you disagree. Obviously, the brain maybe is more fundamental than the body, but we don't have any ways to keep the brain going currently without a body.
Yeah, so I switched specifically to the brain when I was doing my postdoc, and very consciously made that decision that there may be other ways of dealing with the body. But there is, you could replace the body with maybe, a synthetic body, for example, or a biological body, or, there's options there. But for the brain, there isn't really much of an option. It really is who we are. And, we could solve the body. And if we haven't done the brain, then.
Yeah. So before you started working on the brain, I guess there was literally nobody working on brain replacement. Or am I am I understanding that correctly?
Not in the way that would impact aging. Yeah, so there are a lot of labs and a fair amount of companies that are focused on introducing new cells into the brain. Some of those, a lot of those don't make any sense. Some of them do in terms of treating disease, and they've been successful. Cells are very effective treatments if you put the right ones in for the right indication. And so, that's a level of repair, if you like, if those cells are missing or they're not functioning properly. You put new ones in that do function properly or that replace the lost ones, like for Parkinson's, for example, and you can get effective treatments. But but no one is really working on functional tissue for the neocortex. The neocortex is a little more, you have to pay a little more attention to the details for the neocortex because, again, that's the conscious part of our brain. That's what we use for thinking, really defines who we are. And if something's a little askew, you could end up with a psychiatric disease or something else. Whereas if something's a little askew in the cerebellum, well, okay, so you're not a professional athlete, but who cares? You're still, can be doing anything else you want. So there's been a lot of interest, I guess, in neural stem cells and a lot of labs transplanting them in the brain and also transplanting organoids and making organoids. And those studies have been very useful in supporting the idea that these young precursors can integrate into the adult brain. But in every case, the tissue that is derived from those grafts, either single cells or organoids, is completely disorganized within it and is missing a lot of cell types. And so we know from developmental studies in mice that when you have cells that are not organized the way they want, you don't get wiring correctly and you don't get proper cognitive function. So people have been transplanting organoids for over a decade, I guess, brain organoids, and basically seeing the same thing. And everybody's still excited about that. But there's literally been no progress in trying to make those more functional. Why? I don't know. I guess because a lot of the uh, labs working on it don't necessarily understand how the brain normally develops from these early precursors and are missing essential cues to being able to do that. So that's actually what we're working on, is making sure all the cues are there so that we can get structured brain tissue that's developing in the adult brain with the right cell types and will be wired correctly internally in the graft as well as with the rest of the brain.
Hmm. So how does that work? Like, if you can give us the high level step 1, step 2, step 3, how do you replace the brain to save it from aging?
So the replacement process itself,
It's kind of like a high level.
it really is like the ship of Theseus analogy, where you have to do it progressively to allow thoughts, functions, personality to move from one part of the brain to another. And that has to be done slowly enough to allow plasticity to seamlessly re-encode those functions. But yeah, I don't know how much like detail you want in that process. So you can break it down into the removing old tissue, adding new tissue. I don't know.
Yeah, I mean, we want to get into the technicals here on our podcast. That's one of our differentiators, is to get into the weeds on this stuff. And maybe as a sort of starting point, like there is the natural example of benign brain tumors where people's parts of their brain become filled with non-functioning tissue, but they don't, from the outside, seem to have lost any function. And so that's one mechanism by which we know that you can make space for new tissue potentially. And another is that the brain actually shrinks with age, if I'm correct. Although I've heard a lot of different numbers on that. Maybe those would be starting points for like, how could you actually replace parts of the brain? Those are natural examples of like how space can be made. And then also, how do you build these graphs for the cortex? And then also talking about other parts of the brain as well, because obviously they'll age as well. Get as deep as you're comfortable going here. That's what we want to hear.
Okay, there's a lot in that question, but yeah, no, you could remove existing tissue without loss of its content by mimicking what happens in cases of benign tumors. So from a pinpoint out, you get destruction of the tissue while the person is using those functions. Over time, they do not get lost. They just get re-encoded. We would not use a benign, and to be clear, we haven't started working on the silencing of the activity of brain tissue from a pinpoint out. But we do know that technology likely already exists for that. Optogenetics, which is commonly used in all neuroscience labs, where you use particular channels that are responsive to light to shut down the activity of neurons or activate them. But in this case, it would be to shut them down. And because you use laser light to activate these channels to shut down the activity of the neurons, you can do it like a tumor would from a pinpoint out over time and control that rate. And that way, you're not at risk of a benign tumor becoming a glioblastoma, for example, and killing you. So we haven't started working on that, but I think it doesn't require necessarily invention of new technology. It would need adaptation of existing technology for that purpose. And so that's how we're thinking about removing old tissue progressively as we add new tissue. And for the adding new tissue for the neocortex, for the adding new tissue in the neocortex, again, we're letting developmental biology. So the way our cortex was made, the ones we have in our skulls now, no scientist or engineer made them. There's an inherent program in those early fetal precursors that allow for that development to occur. Not only allow it to occur, but what these other studies have shown with dissociated cells that have no structure or organoids that have very abnormal structure is that they still try to do this, and they still try to follow their developmental program in the adult. And they do so successfully in the sense that they do give rise to the right type of neurons that project to the right places and connect. It's just the tissue itself is a jumble. So the way to address that is to have a deeper understanding of this early fetal tissue and how it's made and just mimic that. And so what we've done is high resolution single cell sequencing analysis of the cells that are there in the early human fetal neocortex, as well as a proteomics analysis of the structural components and signaling components around those cells. Even early on, there's at least a couple of layers of precursors that are very important there that are surrounded by completely different proteins. So reassembling those and allowing them to do their thing is the approach, so, as they would in the fetus.
So you're like collecting three-dimensional data of what these things look like at different stages so that you can tell, then you have like you can compare
Yeah,
that to the graphs that you're building and see if they match.
yeah, we're focused on one particular stage that we think is the simplest we can do without losing key components. Um, and yeah, because as cortex development proceeds, it gets complicated relatively quickly, and so we want to make sure that we're building something that we can put together in a way that will still work.
Yeah, so build the least complicated tissue possible that would still complete its process on its own.
Yeah, you had a third question too. No.
Well, I want to highlight too, like you already mentioned that there's the silencing aspect, and then there's other parts of the brain other than the
Oh, other
cortex.
parts.
And
Yeah.
it's like very important that we're working on these things in parallel, right? And so you mentioned people are using technology that could be adapted for silencing, but that there nobody's currently actually working on that. If we're going to solve this problem of aging in our lifetimes, it seems to me that we really need to be working on all of these things in parallel. If we do it
Yeah.
serially, that's going to be too slow. And so,
Yeah.
but that's just an aside. I can let you get back to the.
But yeah, I totally agree. Yeah, the other parts of the brain are essential to function and also to who we are. The neocortex itself doesn't have certain modulatory types of neurons like dopaminergic neurons, serotonergic neurons, um, cholinergic neurons. And so if we were just neocortex, it would be a very robotic existence. Really have we wouldn't be able to attribute weight or feeling to what we experience. So those parts of the brain are also very important. They're not necessarily conscious parts of the brain, so as long as they're wired correctly, um, they may do their thing. So it may be possible to replace them, less like ship of Theseus and more, in not whole or closer to whole than you would with the neocortex.
Yeah, like there's a lot
But
of different parts of the brain. And I mean, everybody, I think, knows that the brain has two hemispheres. It sort of has a bilateral structure. And a lot of the parts other than the cortex, do you think they could replace sort of one half at a time sort of thing? Which parts of the brain do you think that would be possible? And which parts of the brain other than the cortex do you think are more complicated than that?
yeah, I mean, most parts of the brain are pretty complicated in their structure. I don't know that they're any less complicated than the cortex. They don't have a nice laminar organization like the cortex necessarily, but if you think of the hypothalamus, for example, it's a very basic part of the brain, and it has smaller numbers of neurons that do things, but those things are super important. They control your state, your activity state, whether you're hungry, sleepy, maybe we don't need those things in the future, but
No, I
currently
guess my
they're
question is more like, could you replace half of it at a time, and would that, hypothalamus be sufficient temporarily?
I think that's the correct way of thinking about it. You would do half at a time.
Where would we get half of a hypothalamus? Would we have to engineer it from graphs?
Yeah, either engineer it or, the alternative. So we're engineering neocortical tissue, but the alternative would be, collecting it from early fetuses at a stage where there's no neurons yet. So again, there's no sentience, there's no, there's no personhood, there's no person. But that tissue could be very useful. And that could be, different sources of this fetal tissue could be like ex vivo. So, we know companies are working on growing fetal like structures ex vivo.
Embryoids at Renewal Bio.
Yeah. We should be clear. This is all non-sentient.
It's an important part, and I think this is where the overlap between the brain replacement and the non-sentient cloning comes in, right? Because in theory, if you're growing a non-sentient clone, it will still have to have some parts of the brain. And this is actually something that came up in conversation recently with me, which is we know there are examples of people that are born without a cortex, right? There's hydranencephaly, I think is the term. I'm not sure I'm pronouncing that right. It's a tough one. And sometimes those people are kept on life support. But two questions related to that. Would certain parts of the brain, could you just get them from the non-sentient clones? And the second part is, will the peripheral nervous system develop properly in a body that doesn't have a cortex?
Yeah. I think the answer to the second question is yes, probably. Again, the problem is those cortexless human bodies that have been kept alive are usually pretty inactive. And indirectly, that might affect development of muscles and sensory neurons to those muscles. I don't know, but that could, but
That
that's
actually
something.
seems like a project that somebody could do, is because some of these people actually exist. We could actually test it potentially.
Yeah. But, the peripheral nerves have some capacity for growth on their own. And certainly, I think if the body then becomes active with physical activity, muscle builds, that, at least the motor nerves would probably adapt, adjust sensory ones as well, maybe. Yeah. But yeah, I mean, I think the peripheral nervous system at least develops pretty independently of the brain.
I guess I was wondering if there's actual research on that. Have people actually looked at these hydroencephalate patient, like people and bodies,
Yeah.
and seen what the peripheral nervous system looks like?
Yeah, they're not very common, so I don't know that anybody has or would have. I would definitely think that in the mouse literature, you would be
Mm.
able to find examples of that because people have been knocking out this and that, and there's every combination of cell type and gene.
All right,
There, yeah.
good. Somebody listening to this should do that experiment if it hasn't already been done and find out. Speaking of parallelism.
Yeah, there are different cortexless mice out there. So it's probably known already that, my recollection, even with the limb 1 mice that can be born without a head, is the rest of their body is normal. And I would assume that includes the peripheral nervous system. And all it would take would be to see movement after birth. I mean, they don't live long, but if they move at all, that would be indicative, or even pre-birth. Yeah.
Okay.
If we want to, yeah, continue on our devil's advocate train. So in a previous episode, we interviewed Reason from Repair Biotechnologies slash fightaging.org. And when we asked him what was solving aging, I believe he said something like, extending lifespan without requiring major surgery, which I think he was, yeah. Anyways, which I guess includes cryo, but also includes potentially, a head transplant or progressive brain replacement. So maybe we can get a little bit into that, because I think one of the reasons why maybe other people who are working on more traditional pharmaceutical approaches, or even bioengineering approaches, is that they are afraid of this idea of doing any kinds of surgery. It looks just like the solution just looks so different from the typical types of medicines that they're familiar with or comfortable with, right? Like most people don't like surgeries at all, right? So I'm curious what you think about that, because the other thing is there's going to be a risk every time you do surgery. There's going to be a risk, and if you do serial surgeries, those risks compound as well. So I'm wondering if you've thought about how to mitigate these risks to, I don't know, to allow serial sort of procedures and stuff like that.
Yeah, I mean, I'll start by saying surgery has been, probably historically the most effective way of treating ailments, and, certainly in any curative way. And brain surgeries are going on every day in every major hospital around the world. And to say we can't use surgery is just sort of, you know, why not? Like it's sort of standard practice already. So to not want a treatment, to exclude a treatment because it involves surgery is very bizarre. And yeah, I mean, brain surgeries do come with the risk. Others probably more so than brain surgery. I mean, brain surgery, the risk is damage to the brain, but you can actually usually survive that, but you don't want that damage. So I think there's ways of mitigating that. There are technologies being developed that are consistently reducing the invasiveness of brain surgery and therefore the risk associated with brain surgeries. And that's just going to continue getting better. An example that I was really surprised by because I didn't think it was even being used clinically yet was the implantation of electrodes for deep brain stimulation. That's done by a robot, at least in some places, including where I work, and I didn't even know that. And so the technology is really progressing there. It doesn't get much press. I don't know why, but I guess maybe there's not that much money to be made there. Drugs are way more profitable than surgery. And you know, I think that's probably a major reason why we don't hear so much about surgery as effective treatment, even though it is exceedingly common practice in hospitals. Yeah.
Yeah, and one other thing that you just mentioned, this made me think of this, is just like there is like an economic incentive, a force pushing towards improved brain surgery techniques, like in the sense of like BCI. So, all the Neuralink-esque companies, I'm assuming the implants don't last forever, so you'd have to do some serial procedures, or at the very least, there's a huge incentive for them to increase the safety of these procedures, like to the point where most people feel like it's like very safe, like on par with laser eye surgery, where most people aren't too worried about the
Yeah.
complications there. Yeah.
I mean, that's a good example because that's something you're like, oh my God, like a laser in my eye. Are you kidding me? And then it's like, okay, yeah, everybody has it.
Yeah, I
Yeah.
think if the Overton window shifts over time, the types of surgeries that are done on heart, like my now deceased grandfather, I think 40 years ago had an experimental bypass surgery done in his heart. He was actually part of a clinical trial, and they expected him to live 1 year after it. He ended up living 32 years after it. Um, and it's like,
Yeah.
but it was extreme. Like they had to lower his body temperature to extremely low levels so that his brain wasn't damaged while they were doing the operation. Um, so, and another thing, I guess another object, people, yeah. So there's the drugs are more profitable. I think that's obviously one of the issues that replacement faces, not from a technical perspective, but just simply from a investor perspective, which is frustrating because we should be optimizing for reducing human suffering rather than capital efficiency, in my view. But it's just the world we live in. Uh, but the scale up is another thing that people often mention when I mention replacement. It's like, well, how would you scale this up? Like, you can't just manufacture a pill. Like, how are you going to actually make this? And I guess one of my counter arguments is, well, we raise something like 8 trillion animals per year, like extremely cheaply. I mean, we can probably figure out how to grow replacement human parts extremely cheaply if we wanted to. But
Mhm.
do you have any thoughts on the scale up, I guess?
Scaling of tissues, organs is something that is not solved yet, but it's something that the field of regenerative medicine is working on. We're working on it as well. There are trailblazers out there that are doing it for iPS-derived single cell types, and automating that and scaling that, and are being successful at that. Tissues are a little more complicated, but there are companies that we've talked to, um, again, because this is something we're going to have to do as well, that are very interested in working with us. They're already trying to automate the production of 3D tissues. They haven't worked with the brain yet, but the same principles of automation can work there as well. So if we can automate, we can scale, and we're good. And again, these are not necessarily complicated processes. biology is always a little messy. Like when you're working with iPS cells and differentiating them, you don't always, the same protocol doesn't always work the same way for the different cell types or for the different iPS cell starting cells. And so adjustments always need to be made and the process always needs to be monitored. But that is something that some companies are, developing AI for effectively to help in the automation of that. So, I think that's definitely solvable that way.
Yeah, I've spoke to two companies recently that are working on solutions to that. Um, so yeah, there's Frontier Bio. There's another one that I think is maybe not public yet.
Hmm.
Um, so I guess I think there's one more major area of concern that people usually bring up to me, which is the nerve reconnection part. Because, for instance, spinal cord injury has been a long-standing area where people have been trying to come up with treatments for it, and there still isn't a standard of care for that is reliable. Although there are some interesting technology demonstrations, I would say. But I guess what to you, I'd like to get your thoughts on like what research areas are interesting, what labs are interesting, what companies are interesting, and yeah, do you see any major scientific issues there still outstanding, questions, or is it more of an engineering problem? Yeah, what are your thoughts?
I mean, for the spinal cord, there are some, you know, tantalizing findings, newer findings showing that you could potentially engineer a fix to it without replacing spinal cord tissue. Um, I don't know why, but no one has really focused on, you know, growing new spinal cord tissue the way that it normally develops. There's literally no one working on that. Everything but that, it seems, for the spinal cord, and yet to me that makes the most sense. How is spinal cord tissue made? Well, we don't. Let's just make it the way it's normally made. Um, so maybe somebody will work on that at some point, but even without that, the spinal cord, you could potentially get a good enough fix without it being normal, as long as, maybe it's through a relay instead of, continuous axons. Um, yeah, so I think that's
I think there's a
poten-
is there a company called BlackRock that's doing a BCI like relay to
oh, yeah,
bypass the
yeah,
injury?
right, right. No, there's, yeah, yeah. That's one. Yeah. Um, and they've had success. I mean, you could say Neuralink is doing the same thing, right? Their first indication is like quadriplegia, I believe. And they're not the only two. There's at least another one that was doing trials in Australia already. They were the first to do trials, I think. I forget what they were called, but yeah. So you can bypass the spinal cord as well. Um,
Inducing healing, I think, is another area too. I mean, there was the Sinclair paper on optic nerve injury and then reprogramming. Although I
mhm.
don't think that was a complete severance. Although I don't know the details exactly of that experiment.
Yeah, they crushed the optic nerve.
Yeah.
And I think that actually brings up an interesting project. ARPA-H, I think, has approved a eye transplantation project. Do you know any of the details about that? Because that seems relevant, right? Because you're going to have to also replace the sensory organs. Uh,
Yeah,
so
yeah. No, that's I was very happy to see that. Um, there's a word there that's really important is functional eye transplant, because eye transplants have been done, before and for aesthetic reasons, right? Just to replace the eyes, but they're not functional. So this would be a functional eye transplant. So that's, I think, that's an encouraging, um, you know, news from a government entity, that they're willing to tackle these more ambitious projects that, people really, I mean, scientists, really don't have recourse otherwise, because the typical government funding mechanisms don't do anything big like that. So it's nice to see ARPA-H in place doing these things.
Yeah, I think we've been really excited for the potential of some longevity minded people getting positions in ARPA specifically so that they can direct funds towards hard technology development problems that are otherwise maybe too ambitious or too outlandish for government or VC. Um, and
Yep.
I take it that you're also considering a position at ARPA H. I don't know if you have any comments
Yeah.
on the general idea of government, of how to get these ambitious ideas funded, and also of your
Yeah.
potential.
I submitted a program proposal to them um, on cortical tissue replacement. In fact, at first I thought that would be beyond their interests, too specific, not appealing to them or the general population. So I had entitled it, functional brain repair or something. And they pushed me to call it cortical tissue replacement, because that's really what I was proposing. But to their credit, they didn't shy away at all, and they really helped in my discussions with them to make it clear and ambitious, and and to get it to work. Like, what are the steps that would get it would get us there through an ARPA-H program. So yeah, it's a very attractive proposition. Um,
Nice, yeah. I'm hoping that you or other people like you end up in those positions because it is funding is a major constraint for these problems, and it would be like we've said a couple of times already. There's multiple things that need to be funded in parallel here for us to actually optimize for time to getting solutions to like the whole problem of aging.
yeah,
Uh,
One thing to bring up, since Mark mentioned time, and I think this is really important, right? The fact that time is ticking. So there's this Tim Urban post, right? Blog post about your life in weeks. And there's a website that I encourage people to check out, where you can just like Google like your life in weeks, and you can put in your birth date, and it'll print you a little chart of like how many little squares representing a week in your life in a typical normal lifespan you have remaining. And when we're in the context of the roadmap, one of the things we wanted to do for the LBF roadmap, technical roadmap to solve aging, was to get estimates on time that it would take to develop these technologies, and also cost in terms of money and resources. And for some of the strategies like bioengineering, it's just like it's so open ended. We it's really hard to even estimate how much it would be. We know it'd be like it could be like trillions or more, right? But when we talk, the interesting thing is when we talk to people in replacement, um, they're more the timelines there are more constrained, or at least definite. There's still going to be some leeway, and also because of the timelines being definite, the costs are also somewhat more estimatable. So I don't know if you wanted to speak to that, just like how much you think it would cost to in terms of time and resources to get to like a, I guess, proof of concept. We'll put away like scaling to the side for now, but just like proof of concept to test whether this strategy will work. I mean, yeah, there might be some unforeseen sort of things, but
yeah.
I don't know. Maybe you want to talk to.
Well, I mean, with your roadmap, you guys are now the experts on that. So, I can reference you guys because you've talked to more people, you've asked more people, this question than I have. But no, I mean, I totally agree with what you're saying. It's the replacement approach is a much more defined strategy, and we know what the milestones are, and we know how to get there. And so that also, helps define the cost to some extent. I think the cost is maybe more more defined than the timeline, because if we don't get the money, then the timeline doesn't mean anything. We just won't get there. So, I threw out $1 billion for replacement to beat aging. You guys came up with, I think, 3.6. Yeah, whatever. Let's say 10. It's still not that much, right? And I think most of that has to go to the brain. I think the body is pretty, much. None of this is easy, but the body is an easier project than the brain in terms of replacement. Yeah, and I would put the brain at tenfold more complicated than replacing the body. So give it 10 times more funds.
Yeah. I think people are shocked when you hear these numbers, because it's like, $10 billion, like in the grand scheme of things, that's
Yeah.
nothing. That's like a rounding error on the amount of R&D that's spent on pharma research.
And I've heard the argument, oh no, it's going to be, just without defining what the approach to beating aging would be, it'd be like, oh no, it's going to cost trillions of dollars, because we don't even understand what is causing aging yet, and there's so much to understand. And I'm like, yeah, okay, well, you're obviously not thinking of replacement. We don't need to understand the thousands of things that are going wrong if you're just replacing. So,
yeah. I thought it was really funny when we published our roadmap and people read that estimate, that $3 billion estimate. They accused us of being too optimistic, and they were just like, "Oh, you're definitely underestimating the complexity of aging." We're just like, "Well, replacement, you just don't need to know.
Yeah,
You don't need to
exactly.
understand the complexity of
Exactly.
aging, and that's why it's
Yeah.
And I thought that comment was funny because Mark is a known pessimist.
Yeah, and even to the degree that you do need to understand aging, replacement allows you to bypass at least some portion of it, right? So it's definitely, it seems like, just from a logical perspective, you're going to need to know less about aging than you would if you choose to do it without replacement.
And I think one other thing is, for clarification, this $3 billion or $10 billion, whatever you want to call it, this estimate is not a guarantee that we will solve aging if you had $10 billion. It just means that you can test out and develop the hypothesis to get some sort of readout to figure out whether it works or not. And there could be unforeseen
people spend a
why.
lot more money than that on much less plausible things, on pharmaceuticals, I would say, currently.
Yeah. Still going.
So this brings, I think there's one more major pillar to the roadmap that I think you also have an interest in, which is biostasis, which would mostly consist of reversible cryostasis, but there are other potential strategies there. So maybe start with a high level of what you think of that approach.
Yeah, I think it's definitely something we should be pursuing. For me, it's a plan B, and I think for most of us, probably it's a plan B. The scariest thing about that for me is the trust that that the vat will be taken care of for however much time is needed, and the technology will continue to be developed for bringing us back, which is why I'm really focused on plan A. But yeah, no, I think it's definitely something of interest that I'd like to see developed also in parallel. I mean, be nice.
Yeah, the major, I guess, are you currently signed up for Cryonics?
I'm not. No.
Are you, Mark?
I'm not also. Are you, Nathan?
I am also not, but that's mostly out of procrastination. I have a life insurance.
But I talked to one of the leaders in the field, Emil Kenzura, right? And, I don't want to mess with his business, but if we're young and healthy, we're more likely to die of an accident than a disease. So if you have a disease and your days are limited, it makes sense, right? But otherwise, the business is not so widespread that they could respond in time, after you've had a car accident or something, wherever that is. So I guess it makes less sense. But still, you never know. And it's an insurance policy. Maybe we're rationalizing. We should all sign up.
I think we should, and I think that anyone listening to this should as well, assuming they can afford it. And insurance is not necessarily very expensive, although it depends on how old you are and other situations.
Yeah.
But for me, I guess the current state of the art is sort of when you weight it against that also lowers my sort of incentive structure. Like, to me, with the current state of the art, your chances of being revived depend on some very hand-wavy arguments about future nanotechnology, because we know the amount of damage done during the rewarming process. For something the size of the human brain, it doesn't seem like you would be recoverable without some form of level of technology that we would consider magic, I guess, at this point. And it's hard to even imagine what it would look like. But at the same time, cryo has been a chronically underfunded field with a lot of very promising ideas that just haven't even been tested, or certainly not tested rigorously. So I guess, is it an area that you've had time to look into the technicals at all? And do you have any particular concerns or projects or approaches that you think should get some funding?
Yeah, I mean, it'd be great to see an ARPA-H project on that, and that would definitely should be on the radar of anybody interested in an ARPA-H position. It would be if I move forward and become an ARPA-H program manager. I recently spoke to, a couple of experts in the field working on cryo, and it was enlightening. But along the lines of what you were saying, there is just so much that hasn't been tested yet. Even basic stuff like cryoprotectants. It's basically a small fraction of potentially effective cryoprotectants, and some could be a lot better than what's being used today and would have the properties needed to be able to freeze and thaw without damage. And that was just a starting point. Then there's also the different technologies for heating, rewarming, and for vitrification, fast cooling. So yeah, it is a really underexplored area as well.
I think cryobiology too, there are organisms that exist that can be frozen and thawed, and they're perfectly fine. So again, there's a proof of concept in nature, which means that we sort of know that it's not impossible. We just and it's a question of how difficult would this be to implement for a human. And so, knowing that it's possible, why not do a hard push on solving those, adapting it to humanity? Yeah, that would be my thinking.
So there's another aspect of, let's say, biostasis that maybe touches on brain replacement, because there's cryopreservation, but then there's a whole other faction of people within, let's say, biostasis preservation, who think that cryopreservation is too optimistic already, and what we should be doing is chemical fixation, because then that ensures that all the structure is preserved if you use, sort of aldehydes or different chemical fixatives. And there, a lot of people who believe in that strategy think that the way out is essentially destructive uploading. Like there's no way to undo these crosslinks easily in their perspective. So you'd have to essentially, scan the brain and emulate it, I guess, on a computer, which is in some sense sort of like a type of replacement. It's like synthetic replacement, going off the biological substrate. Instead of neurons, you're using silicon. What do you think about that? Or is that too out there?
No, I mean, if you, I think that's just a copy of you. It's not you. And if you care about you and you want you to continue, you basically killed yourselves by not fixing yourself in a way that's reversible. And I guess you can try to do thought experiments along those lines. Like if you could upload, and you were still alive, for example, if you could upload and copy your mind to another substrate, or would that be you, or would that be a copy? If you were still alive, you could easily test that, right? You could destroy the copy, and would you be any less conscious, any less you? No. And you can make 10, 20 copies of you. Would you feel more alive, more you? No, you wouldn't feel any of that. It's not you, it's a copy, right?
Yeah, my intuition matches you on this. Like it gets into philosophy, and it's not really, maybe it's not a scientific question per se, but I would say the plan A is to keep the original instance alive. But yeah, I don't know. There are some people that seem to think on philosophical grounds that actually it is you. I don't understand their arguments.
Yeah, people don't seem to care. It's like, well, if the copies,
I think that seems to be the case, is that they don't actually care, maybe, or maybe they just or operate under a totally different framework of thinking that doesn't make sense to me. There's always been a gap there that I don't seem to understand.
Yeah, but I also hear that it's like, well, we don't know what consciousness is, so that could be you, but unless it's if it's part of the physical universe, it's not something that, it's not like a spirit that, can go from one substrate to another. It's embedded in the physical world, which happens to be our brain in this case. So yeah, I don't buy that argument either. Or maybe consciousness is this thing that is not
independent somehow. Yeah, it seems like you're drifting into, what is it? Is the consciousness being separate from materialism?
Yeah. There's a word for that.
Okay. So I think get into some final thoughts here. We want to get, we're trying to give advice to people, right? Like ultimately the goal of this podcast is to help people make decisions on what they work on, what they invest in, how they spend their time and their money. And so a few questions along those lines. I'll start with one, which is, who are people that should be followed? I mean, other than yourself, I definitely recommend everybody read your book, even though, as you say, it's maybe not completely up to date. It's a very good book. Who are other people that you think entrants to the field should follow?
Yeah, I think there are a few critics out there of the longevity field in general I think they're worth following because they usually make their arguments based on science. They're not necessarily fans of replacement or know about replacement or endorse replacement. Um, you know, uh, I mean, they're kind of out there, but they're worth following because they tend to be science based, and no one tends to listen to them in the longevity field that much. But maybe they should because they are.
Who specifically?
I mean, I can think of like Charles Brenner now. I mean, maybe he's not like the most, maybe he's looking a little bit for a fight. I don't know, but he does make good points. And maybe he's right to look for a fight to some extent because he finds a lot of what's said in the longevity field misleading, and I would agree with him. You know, yeah, there may be a couple of others, but yeah. Otherwise, anybody working on replacements, I think, is worth following. Probably people you've had on, your podcasts or you've interviewed.
Yeah. Well, we definitely have a list on our website of recommended reading, and the authors of those publications, papers, articles, books are people that we recommend following. There's also the series of foresight talks as well, I think are interesting. Another question is, do you have advice for the LBF, for us, for how we could be more effective? You've been following us. I'd love to get your feedback on what we're doing. Oh,
This phone probably died.
Yeah, it's just like, it's a waste of time. Why am I even
here? Probably when you ask them about feedback, he's just like, you guys are beyond saving.
Yeah.
It's just like, I'm out of here.
Yeah.
We'll see if he comes back.
No problem. We're almost done. Just want to grab a couple more.
I plugged it into an extension cord that had a bad connection or something.
No problem.
Maybe you should replace that extension cord.
Yes, exactly.
Progressively, or maybe all at once.
That is the simplest solution.
Yeah, instead of opening it up, and why did it fail?
Yeah, can we unoxidize this metal?
Yeah.
That's actually not like, this is more relevant than you think. Like nuclear reactors are like one of the most complex systems that we've engineered. And whenever like you try and like repair it, right, it's always about like just replacing the tubes and stuff like that. We don't actually go in and try and like add chemicals to like remove the rust or anything. It's just way simpler to just replace all the bad parts.
Yeah, same with cars, right? They just replace parts.
Yeah, or just buy a completely new car. And transplant the person into the new car.
No, we should fix cars by inventing molecular nanotechnology that can go inside and
or we put it in liquid nitrogen, keep it there until we develop the technology to slow car aging.
Yeah, I hope this is being recorded.
Yeah, it is.
Yeah, it is. This will be in like the outtakes.
Yeah.
All right. Yeah. So I guess just to return to the question, you've been following Nathan and I's career and trying to get more people involved in the field, and obviously trying to get more people involved in certain areas like replacement and cryo and certain aspects of bioengineering. How do you think we can get more people? Like, what's your feedback for us? How could we be doing better at getting more people involved? Or just in general, how could we get more people involved and more money involved in this?
Yeah, I think the last thing you said there is probably the most important, getting more money, because I think the talent is largely out there. Regenerative medicine, neuroscience are like two of the hottest things in biology. There's tons of talent, tons of young investigators out there studying these things because they're the most exciting. They find them the most exciting part of what's going on in biology, biotech, health medicine. And so it's really, getting those people attracted. Basically, they need to get paid, right? So we need the money to pay them. I think it's also good just to make these concepts, these ideas, beating aging, more mainstream. The more people find this, the better. Okay, the more people are going to be like, okay, well, let's do it because I'm getting old. And, my family's getting old, my parents are getting old, we have to do this soon. So I think getting more people involved in that sense, thinking about it and seeing that there is a way of getting there. Not entirely clear how long it's going to take yet, but there is a way of getting there. I think that's important too. So I think you guys also play an important role just connecting us. I think a lot of the time I've met a lot of people through you guys. I think a couple of those people ended up in my lab. So in a very real, practical way, you guys have helped that way as well. So thanks.
Any thoughts on how we can get more money into the space, specifically the more hardcore, ambitious moonshots for beating aging?
Yeah, I'm afraid it's going to take proof of concept. So we're going to have to grind with what we have, do more with less, and achieve proof of concept that will then tip the scale. I mean, we'll get to a point where it'll be the fable of the dragon tyrant, but for that, we'll need some proof of concept.
Do you have a sense of what that proof of concept might be?
I think for the brain, it's showing functionality of the grafted tissue. I think that's a game changer.
Primate or in human or in...
You can break it down a little. So if it's a preclinical study, it might be, okay, great. You can regain this visual processing in a way that's functional and allows the animal subject to perform a task that it wasn't able to before. Fantastic. But that's not human. That's not what we care about. I think that's still a huge, important proof of concept that should carry a ton of weight and should make life a lot easier for us in terms of making progress. But there might still be some resistance. So ultimately, this being done in humans is what's going to, I think, completely tip the scale.
Yeah, like the pharmaceutical industry has a huge history. And so people have a sort of like understanding of it and a comfort level with it. I think that's a big reason why we keep on pouring more and more money into that versus novel approaches, because people are just reluctant to try new things, unfortunately. Even when the old ways are obviously failing. Yeah, you mentioned people. We've definitely directed talent and attempted to direct resources towards your projects. But yeah, what do you tell our listeners? How can we help you? Do you need any particular types of people you're hiring? Are you looking for funding for any particular projects, whether it be academic or private? Any particular tools, services, volunteer labor that could be useful? What could help you move faster?
Yeah. I think it's really funding. I don't know what I don't know in terms of connections. Maybe there are people out there that I haven't been connected to yet through you guys. But I feel like I know a lot of people already because of you and other people that are facilitating this. So there's nothing like, oh, if only I knew somebody who could do this, or if only we had this technology. I would just say keep educating us on the state of the art of some enabling technologies like automated cell or tissue manufacturing, like those kinds of companies, because we're going to want to work with them. There's potential for a lot of interaction between many of our companies. So yeah, just keeping us abreast.
Yeah, there are two companies that I'll connect you with that I think you might not know about that are working in that area.
Yeah.
And then,
Frontier Bio. Yeah.
No, the two other ones, actually.
Two other ones. Yeah.
That I just, know about more recently. So volunteer, is there anything that volunteers could do? I guess raising awareness of your work. I think one thing is that,
Oh, yeah.
like,
I mean, if they're, if they do have like stem cell training or neurophysiology training, surgical training, and they're willing to help, yeah, we'll take it.
awesome.
We might not
almost
be able
view it
to
as
take-
like apprenticeship, because there's not many people doing what you do, and so expanding the number of people that understand and know the things that you do seems useful. For the audience, any investors out there, do you have any currently investable projects?
Do we? Yeah.
Okay, tell us.
Yeah. So I have a company, BE Therapeutics, and we're making fetal-like human cortical tissue for the purpose of replacement.
Okay. All right. And so there's an open round right now?
There's an open round. Yeah. The seed round is still open. Terms are very investor friendly. Yep.
All right. Good to know. I'll keep sending people your way, and maybe somebody will hear this and just reach out to you on their own. And how should people reach out to you?
Email is best. Pretty easy to find by email. betherapeutics.com also works. Yep.
Awesome. Well,
great.
Any final thoughts, Nathan?
I think we covered a lot. And always honor to talk to Jean, because your book really changed the way I thought about aging, and he's one of the leaders in the field, actually working on one of the most important paths to potentially beat aging completely. And I always tell people he's 1 in a billion, like literally 1 in a billion people working on actually trying to solve aging in the brain through replacement. So yeah, thanks, Jean, for all the work you do.
Yeah, no, honored to be here with you guys, and
super, super grateful, and we'll hopefully make up for the time you spent by connecting you with more of the resources and people that you need. So, all right. Goodbye.
Always
for
good
now.
spending time. Yeah. See you for now. Talk soon.
Oh.